Welcome to the Purr Podcast, your favorite feline-focused podcast, where we dive into all things feline, medicine, surgery, behavior, and of course, a sprinkle of veterinary gossip along the way.
I'm Dr. Jolle, feline fanatic, surgery nerd, and self-proclaimed lover of all things awkwardly cat-related.
And I'm Dr. Susan Little, proud cat vet, podcast enthusiast, and your guide to the wonderfully weird world of feline medicine. Let's get started.
We are at our second episode, and we had a cliffhanger.
We did.
So what was it?
Yes. Yola always likes to kind of cut things off right at the point where Susan's leaning in and going, yeah. So we're very fortunate to have Dr. Amir Kol with us for part two. Thank you. Yeah. So many unanswered questions. So I think we left off where you were talking about this unthankfully rare condition in children who have had COVID and how that can look kind of similar to cats that get FIP, right? Because it's an unusual, it's like an aberrant thing. And so if I remember correctly, the grant we started to talk about was about that disease in children, was it? And then where's the feline tie-in?
Right.
Yeah.
So, because NIH, they care about human health,
sure.
right? They only care about human health, right? But in order to study human health, they use lots of animal models, right? Typically, people use mice, rats, sometimes pigs, right? Things that depend on what kind of study, whether it's a pathogenesis study or treatment, right? And actually, naturally occurring diseases in companion animals, in spite of the fact that, for us veterinarians, we all know how great of a resource could that be, that it's, real diseases in real animals that have the complexities.
Same environment.
Yeah, same environment, all of the above, right? NIH is still somewhat more reluctant because, they know mice and rats, right? They know that it's not, they know that these are not great models, but they know it. But they know it, right? As opposed to, our weird disease of veterinarian, right? They just, so, I had to, in the grant, I had to convince them that, that FIP is potentially a really valuable model of MISC, right? That was kind of the context, right? So again, they don't care at all about FIP. They don't care about cats. It's not within their purview, right? They care about children, right? So I had to convince them that we can learn from FIP about what happens in kids and potentially, understand the disease better and offer better treatment options.
And can you explain that to us in very short? You don't have to read the whole application that you did, but how did you make that comparison? So why is the cat a good model?
So, both are diseases that are driven by coronavirus, right? So obviously, it's a different coronavirus, but both are coronavirus. Both are really, the hallmark of it is a systemic inflammation, so throughout the body, and an aberrant immune response that can get really severe and potentially deadly. Right?
Yeah, that's where your own immune system becomes part of it.
And I think the one that you forgot that I remember from last time was the fact that the virus is not visible. It is still there, but you can't see it in blood, and
Right, exactly. So the virus is. It's really because, from the perspective of the virus, it's really unusual because the virus doesn't shed. So, it's a dead end from the perspective of the virus, right? It's not shedding. It's not contagious, right? So both the kids and cats are not contagious.
It's not a good survival strategy for the virus.
No, it's very odd.
So it's clearly not meant to do that, right?
Right, exactly. Yeah. So it's like an odd dead end from the perspective of the virus, right? And the host will, from the cat, they'll die. And kids, if they're not treated, they will also die, right? So it's not a good strategy from the perspective of the virus. And it's really just kind of, it's an offshoot. It's something that goes wrong, also from the perspective of the host, but also from the perspective of the virus.
Right. Yeah, that's a good way to look at it. So presumably, especially in the early days of the pandemic, I'm guessing that there was an assumption if you had COVID and you recovered that you got rid of the virus, or was there already some suspicion it might not really be gone? Right. We don't know right at the beginning of anything. We really don't know.
I think, I'm not sure how long did it take him to, realize that there is also long COVID, right? So that's another syndrome of kind of persistent inflammation and disease. And honestly, I don't know enough about long COVID, so I'm not sure whether it's, what's driving it, whether there is persistent infection or not. But in MISC, there definitely is persistent infection without shedding.
So the virus is hiding presumably in some tissues.
Correct.
Yeah. Where does it like to hide in children? Do we know?
Multiple organs.
Doesn't matter anywhere.
Yeah, multiple organs. So heart, brain.
Right. So rather like FIP, like virus will. Right. And so PCR on blood doesn't pick it up.
No.
So how do you find it in tissues? So
in children, there is not a lot of you have to find an autopsies, or potentially if you get a biopsy.
Okay, let's say it's a biopsy.
Right. So if it's a biopsy, you could just you can sample the tissue, and then you could do PCR. You could stain for the virus with immunohistochemistry or whatever. Other
So
they're not viremic.
No, they're not.
Yeah. So that's the challenge.
And parents have told us yesterday that her theory is that it starts in the GI tract and the virus is there, and then something happens in that blood.
Well, it gets into monocytes. She's just talking about that, right?
That's I would say that's the dogma, right? So it starts with regular kind of enteric coronavirus, and then for reasons that are not understood, which is also kind of mind-blowing, the virus undergoes a mutation, but it undergoes the same mutation each time, so it recreates the FIP virus each time de novo, which is like super bizarre, right? Because again, like from the perspective of the virus, it's a dead end. Like it won't infect other cats, right? So then it undergoes a mutation that renders it can infect monocytes and other potential cells that are outside of the lining of the gut, the enterocytes,
and then go anywhere.
It can go systemically, exactly. And then
and we're very happy that it is not mutating in something that can be spread. I mean, let's say that because that's the worst nightmare that we have, that there will be at a certain point a mutation that can do both.
Of course.
Yeah. It's often hard to explain whether I'm doing a talk on FIP or talking to other vets or whatever, that it is and isn't a contagious disease, right? Like the coronavirus is contagious. But the FIP part.
FIP is infectious, but it's not contagious.
Yeah, exactly. Yeah, so it's an odd virus.
It's an odd disease. Yeah. It's a really interesting and odd disease.
So you wrote the grant,
and then what?
Oh, and then I got it. I was like,
it was not that easy.
Not that easy at all. No. It's really, I can tell you, because I've been in this business for a little while and I've seen my colleagues there. It's not easy to convince NIH that a naturally occurring disease in companion animals is a good model, because it's not only that and there are lots of good reasons for that, honestly. I mean, it's because in a model you want it to be predictable, you want it to be manipulated, you want to have the tools to study that, right? And in veterinary medicine, these are naturally occurring diseases. So, I don't know when those cats got infected with coronavirus, for example. I don't know when they present to the hospital. And then we're also lacking lots of tools, mostly antibiotics, right? So, for humans, rats, and mice, there are tons of antibiotics for any kind of, target that you can imagine, right? And in dogs and cats,
it's much, like 10.
Exactly. It's much more challenging, right? And lots of the work that we're doing, whether it's flow cytometry or, lots of it is based on availability of antibodies. Nowadays, in the past few years, we have, sequencing is much more available, and sequencing is cheaper, and it's much more species agnostic, meaning you can sequence whatever you want, doesn't really matter. That being said, in order to interpret sequencing, you need the genome to be really well annotated.
Yeah.
And that's also still, we're still lagging way behind, in cat and dog.
Cat has made a lot of progress, though.
Sure.
Yeah. So Leslie Lyons is a very good friend of ours, and she's been on our podcast many, many times, and we've talked feline genome a lot with her.
Yeah, exactly. She's contributed, a tons, and definitely it's getting way better.
Yep.
But I can tell you, like I'm doing part of the study we are doing, we were able to do something that to me blew my mind. But with collaboration with our radiologists, they were able to get us lymph node aspirates, so mesenteric lymph node aspirates, from these cats. And the lymph nodes are really critical because this is a site that gets infected, right? That's the virus gets there and infects there, but also this is a place where the immune system is being kind of, mounted and educated, kind of formed, right? So it's like a really key organ.
If it goes wrong there, then.
So we were able, the radiologists at our, teaching hospital were able to get ultrasound guided fine needle aspirates of those mesenteric lymph nodes, and then in the lab we were able to basically sequence the entire transcriptome of each individual cell, so single cell RNA-seq, which is mind-blowing, right? You think like 10, 15 years ago, that would have been
that would have been a grant in and of itself.
Beyond the grant. That was like 15 years ago. That was like unconceivable, right?
Somebody's dream, I guess.
Yeah. So now we can look at each individual cell and look at all of the genes that are being expressed in that individual cell, and we have tens of thousands of those cells, right? Yeah. So it's a hugely powerful tool. Nonetheless, like I can see now, it's kind of frustrating because lots of the genes that I see there are not annotated. So I would just get I just get a number, and I don't know what that gene is, right?
Right. Yeah. Once you get down to that level, I guess is when you start to see the holes in the puzzle.
Yeah.
Right.
But, how did you convince NIH to grant you? So what was the key?
Yeah, was there anything you think flipped them to?
So what question did you answer for them that they thought, hey, that's interesting? Because this is also, if you get an NIH grant, it gives you also a lot of credits.
Right.
It's really good for your career. Yeah. In veterinary medicine, there's not that many people that get NIH grants. It's really difficult, you said already, especially when you do comparative research.
Yeah.
I think that I benefited from the fact that it was the pandemic and no one knew anything.
Yeah.
Right?
Timing.
Especially about MISC, right? No one knew anything about it. So they were like, yes.
Yeah. We'll take you.
Yeah, let's try it.
Yeah.
Right? They didn't do anything. It's very different. Let's say that, for example, obesity and type 2 diabetes, right? That's been for a long time. There are lots of models that are very well established, right? If you would like to, enter with a new model to suggest NIH, a new model for, type 2 diabetes and obesity, like they would be really, really, really skeptical, and, you'd have to stand up to a really high bar. I was fortunate again to get into a situation where there was this new horrible disease. No one knew anything about that, and I proposed something new. So they were like, I think that they were like, right, let's give this person a shot. Plus, it was so NIH has multiple types of grants. Mm-hmm. So this grant is called an R21, which is a high risk, high reward. So it's a smaller grant. It's a 2-year grant with a smaller budget than your typical standard, let's say, grant, which is a 5-year grant. It's called an R01. So they were willing to, give me a chance. And it's my job now to show them that, hey, we've done something useful with that, and we want to keep on studying that disease. And, next my plan is to submit an R1 to keep on.
Right. And I think, if you get this first grant and you show that it is benefit, there's a higher chance that you will get an R.
So where are you in that research right now?
So this is what I'm actually going to talk about later today in the conference. So we proposed to do a relatively small veterinary clinical trial where we enroll cats that have naturally occurring FIP, cats that have relatively at the beginning of the disease, so they still have good prognosis overall, and treat them either just with the antiviral drugs or antiviral drugs with MSC, and then follow them up and take a bunch of samples throughout the course of disease and treatment and try to see what their immune system basically is doing, when they're just presenting, when they have no treatment, and then when they are treated either with antiviral only or with antiviral and MSC.
And so now I understand probably what you were proposing to them is that the MSCs will modulate the response, so the chance of developing this FIP, this deadly disease, is less.
Yes. So, the thought was that, we know that by now, we know that the antiviral drug, the GS drug, is really very effective at getting rid of the virus. But especially if you start off treatment not immediately, this disease targets the immune system in a really significant way.
Yeah.
And we have very little. Now we have a little bit more follow-up. I couldn't imagine that, the immune system would recover back to normal, like with no problem like that. So, and now we know that, like, there are multiple reports, now we have from our experience, it's been reported that cats that have recovered from FIP after being treated with a GS drug, they still oftentimes have enlarged lymph nodes, right? Yeah. Indicating that, their immune system is not back to being balanced.
Yeah,
right?
They're not back,
And again, from our HIV experience with, MSC treating HIV and the non-human primate model SIV, we knew that MSC could potentially help regenerating and recovering the immune system, right? So kind of bring it back, bring balance back.
Yeah, so it's not just we know we can get most cats to recover now, right?
Right.
Because of the antivirals. So you're looking at how can we return them to normal? How can we prevent relapses? How can we prevent all this sort of long-term?
Yep.
Okay. Yep. So I have questions.
Perfect.
So
hopefully I have some answers.
Oh, yeah. FIP is a diverse. Clinically diverse disease, right?
Yep.
So the cats that are in this study, did you have criteria? Could they be effusive, primarily non-effusive? Did that matter?
Yeah, absolutely. So, we wanted to start off with cats that start off with, lowest bar possible. So we started off with cats that are effusive, so wet FIP, and not only that they had wet FIP, only cats that were abdominal effusion only. So we didn't want initially to have any pleural effusion, and we wanted the cats to be relatively stable. So at the beginning, of their disease, so that they have higher chances of surviving, right? And indeed, all of our cats survived. So we enrolled 10 cats. All of them survived to the end of that. So they all recovered. Yep.
So the stromal cells came from where? Autologous?
No.
Of course, that would make sense.
So these are, so autologous, there are lots of advantages with autologous, but there are several limitations with that. The one is, time. Yeah. You need to extract the cells and expand them, and it takes time. Yeah. The other thing is that it's invasive, right? Yeah. We can't expand those cells from blood. We have to get it usually we take it from fat tissue, and those cats, they're not candidates for
they don't have a lot of fat.
They don't have a lot of fat. They're not candidates. You don't want to put them to sleep, right? So, and then the other thing that we learned, a few years ago, we saw cats when we were using cat MSC for a different disease, and we were using at that time, autologous, we noticed that many of them actually, when we start culturing them in the lab, after, a couple of weeks in the lab, they would get, the cells would all of a sudden get super big, foamy, and then after a few more days, they just die off. They just don't do well. And then we investigated that, and we figured out, sure enough, that many cats are infected with foamy virus, feline foamy virus, and the cats, they don't suffer from that at all. It's but they do have persistent infection with that foamy virus,
right? Fascinating.
And then when you put the cells in culture, the foamy virus, for whatever reason, kind of wakes up and kills the cells.
That's fascinating.
Yeah, so we created a stock of cells which we screened for foamy virus. We know that they grow well. We know that they don't have foamy virus, and then we could use that to treat other cats.
Foamy virus, you learn about it once in vet school, and then you never...
Yeah.
That's crazy.
Yeah. In some places, we didn't do that, but there are a few reports, in some places up to 70% of the cat populations are going to be infected with foamy virus.
And what is foamy virus?
What is foamy virus?
Yeah, what kind of virus is it?
I think it's a retrovirus.
I think it's a retrovirus, meaning it gets integrated into the genome, and then it can be latent for forever, and it doesn't cause any. Not that we know, it doesn't cause any problem that we know in those cats. No clinical disease is associated with it. but it just stays there, dormant in the genome. And then I guess when you put the cells in culture, something reactivates it.
Right.
Well, it's an abnormal environment, right?
Of course.
Yeah.
Exactly.
Yeah.
So the other question I have is, how do you know that these strange MSC cells don't cause an immune reaction itself?
Oh.
Excellent question. So, they are cells from a third party, right? So, so you'd expect that they would definitely, elicit an immune response, right? A non-self immune response. So, in a nutshell, they do, right? They do elicit, an anti, kind of xenograft, if you'd like, immune response.
Right. Yeah.
But so what MSC, what they do, they secrete tons of chemicals, right? Kind of, bioactive molecules. And many of them, again, as I said earlier, they modulate the immune system, right? So, initially when, the early days of MSC, they thought that they are completely immune privileged and that they can go completely into stealth mode and they don't elicit any immune response. Now we know that it's not exactly the case. They definitely do. So, they're going to be targeted. They're going to be removed by the body, right? They're going to mount like a CD8 T cell response, etc. They'll clear them. And the more treatments that you give, the more likely that, it will happen faster. But what we do know is that it doesn't cause, like, for example, if you give if you give a blood product for red cell that is incompatible, not only that the blood would be lysed and removed, that would cause a systemic, anaphylactic
that you can die in.
MSC, you don't see that.
So that never happens.
That never happens.
Right.
But they will be removed. They will be recognized by the immune system, and they will be removed.
So is it that they have enough time to do their job before? Okay.
Exactly. So you have to remember, while people call them stem cells, they're not really stem cells, and we're not using them as stem cells. Meaning we don't expect them to go and integrate into the tissue and build, differentiate and build new tissue from scratch. What we expect them to do is to go into the injured tissue. They do that. They home to injured tissue, and then they start their, conductor of an orchestra. They just secrete a bunch of factors that are really helpful in lots of contexts of tissue injury, right? Because they are stromal tissue, they are connective tissue, right? And that's what connective tissue does, right? So, for example, they secrete factors that promote new blood vessel growth, right? If you think about it, no matter what kind of tissue injury, you need blood to go into the tissue to remove toxic waste and to bring nutrients and oxygen, right? They, as I said, they modulate the immune system. They recruit local resident stem cells to start and proliferate and generate tissue, right? So they do lots of things that are really useful.
It's pretty impressive.
Yeah. And that's the reason that if you go into clinicaltrials.org and you look at clinical trials with MSC, you'll find tons of different diseases that people are trying to apply MSC because, what they do is not specific to a certain disease, but rather is much more generic to any kind of tissue injury. Right. So people are trying to use MSC for various diseases.
Well, that helps me understand then why, because you do see like a wide variety of things, because there's been other MSC projects in veterinary medicine too, right? And they're all over the place, but now I understand better why, right? Because it's not disease specific. It's kind of like process specific. So you're following these cats long, presumably?
Well, not that long. We've been following them for 11 weeks.
Okay.
So I don't have long-term follow-up on these cats, and that's definitely something that I want to do later on. But for this project, it was designed as an 11-week project.
Okay.
I still think it's kind of paradoxical because you use the MSCs for immune modulation and dampening down but on the other hand, just to
and they're helping fine-tuning an immune response that is more effective antiviral immune response, as opposed to just the immune system is just blasting everything and causing tissue injury.
Could they help with? I mean, that probably is what you're saying. They help orchestrate also the immune cells that they pull in themselves to be more effective against the virus.
That's a good question. So. Could be. We don't know that yet. We don't know exactly how are they doing that. And in my studies so far, we haven't seen, because we work with cats that had good starting point, all cats recovered really well, right? So, it's not as if, cats that didn't get MSC, they got devastating disease, and the cats that had MSC were all awesome, right?
Yeah,
all of them.
Because they all got an antiviral.
All got antiviral,
right.
And they all started off with a relatively good starting point. So the differences that we are seeing are much more subtle, kind of more very, nitty-gritty details of the immune response. But the big picture, we haven't seen so far, any, major difference. So for that, we need to have longer term follow-up potentially, and looking at cats that have much worse, that are more into their disease, right? Cats that have multiple, let's say that they have bicavitary effusion, so cats that have dry FIP.
Yeah, that's what I want to say.
Yeah.
I mean, you took the easy one.
Exactly.
Of course. Yeah. So as you're following these cats, are you blood sampling them periodically, or what's that process?
Yeah, so initially we sampled so they all presented with ascites, right? So we sampled the ascites, and then as long as there was ascites, we sampled the ascites, and then we sampled blood every time that they came for a visit. And then at the beginning, when they presented, we took an aspirate of their mesenteric lymph node for the single-cell RNA-seq, and then at the end, we took another lymph node aspirate. Yep. So
in the blood, what were you looking for?
So in the blood, we were looking for multiple things. So we're just looking at, regular CBCs, right? So looking at one of the things that are similar between FIP and MISC was severe lymphopenia, meaning they have their lymphocytes in the blood are almost like completely gone.
So the disease in children and the disease in cats share that.
Yeah. So severe lymphopenia, meaning they lose lots of their lymphocytes, right? So we were very interested in that, and we were also doing some more sophisticated flow cytometry to understand the composition of those lymphocytes. Who are those lymphocytes?
Who are they?
So, and then we also looked at inflammatory cytokines, and cytokines are mediators, soluble mediators of inflammation. Right. So we were looking at exactly TNF, IL-6, IL-1, interferon gamma, a bunch of them, right? So we,
cool.
Yep. So we were following that serially as well.
Cool. We wish you great luck.
Yeah, and you may have got a foot in the door for more cat. No pressure, but it's got to work, right? You got to make it work, right? For those that follow you and want to get their foot in the door. So this is fascinating to me.
Thank you.
Yeah.
I also like it.
I really, I enjoy viruses.
You enjoy learning about viruses.
Yeah.
I don't think no one enjoys viruses.
No. I always have to qualify that when I say, FIP is my favorite disease. Yeah, you got to qualify that.
Right.
But it's the curious mind part, right?
Yeah.
Why does it do what it does? Wow.
I'm also likes murder mystery. So there's an explanation here. But anyway, this has been wonderful. Thank you so much.
Thank you.
Yeah, thank you for having
the show.
This is the Purr Podcast, where we talk cats, laugh a lot, and learn something new every episode. Together, we bring you the latest updates, surprising cases, expert interviews, and just a touch of chaos, because nothing with cats ever goes as planned.
So whether you're a seasoned veterinary pro, a new grad, or you're just here for the cat puns, you've come to the right place.